thank you for coming. And we do this regularly to try to expose you to famous scientists, to our own scientists who might not be as famous, but all doing interesting work around nutrition and the future of nutrition research. Our institute works on understanding why people differ so much from each other in nutrition research, and nutrition and metabolism. And because when I was trained as a physician, everybody was the same, but now science is telling us that there are quite large differences between people, and understanding those metabolic differences can help us understand where people need a little more of one nutrient versus the other. And that's what we're developing at our center here. And we're very happy to be here, and we're pleased that we can reach out to some of the people who live near our center and tell them what we're doing. Today we have a really special opportunity in that a very famous scientist, Dr. Bruce Ames, was in North Carolina to speak at an international symposium, and because he's a nice guy and because I asked him, he said he'd come down here tonight and talk to you about what he's working on. And Dr. Ames is started out working at the NIH and then moved to the University of California at Berkeley, and he's a famous professor at Berkeley. He started out working in bugs and microbiology, and then he, after about 10-15 years, said, I had enough of that, and switched into cancer research. Developed in those first, when he was a young guy, some really big contributions. A test called the Ames test has been used to discover cancer-causing chemicals. He then, after working a little bit in cancer, maybe 15 years or so, said, I've had enough of that, I'm going to learn something new, and went into nutrition. So it's my luck and all of our luck that he spent a good piece of his career thinking about nutrition, metabolism, aging, and he's going to talk to us a little bit about that tonight. I think that what I like about Bruce is that he's an out-of-the-box thinker. And so he's willing to think things that many of us are afraid to think. And that's the only way you discover really exciting new things, is really to be willing to take the risk and have everybody laugh at you because you've got some idea, but then prove it's right. And so he started doing that with the Ames test. He says his father did that, but don't believe it, he did it. And he's gone on to make many discoveries. He's a member of the National Academy of Science, really a very distinguished honor to be elected to the National Academy. And he's going to talk tonight a little bit about his research on nutrients, micronutrients, and chronic disease, longevity, and I think you're going to find it fascinating. So Bruce, you're on.
Thank you. Thank you for the very nice introduction. My father would have appreciated it, my mother would have believed it. So I'd like to tell you why I think when you're short of any of the 30 different vitamins and minerals that you need to run your metabolism, it ages you faster. So let's see, first slide. So this is your metabolism. And metabolism, biochemists have worked all this out over the last 50 years or so. There are all these pathways doing various things. The cell is incredibly complicated. There are 20,000 genes, and they make 20,000 proteins, and they're mostly enzymes that are doing all this work. How to create energy, how to, anyway. So what do you need to run this factory? You need fuel, and that's fat and carbohydrate, calories, basically. And then you need something else. You need about 40 different micronutrients. And those are the essential vitamins, the essential minerals, some essential amino acids, and a couple of essential fatty acids. Our brain is 30% in the marine omega-3 fatty acids, which are required. And then choline, due to Steve Zeisel here, who's been the pioneer in that. Anyway, if you don't get, so are we getting enough of these things? Well, I'm going to talk about the whole group of these, except I'm not going to talk about the 9 or 10 essential amino acids, because they're in all protein and they're a different kind of regulation and everything. So, and just for rounding off, let's call this 15 vitamins, 15 minerals, and plus a few hangers on here. Are we getting enough? Well, the answer is a great big no. So the committees set up two numbers. The RDA, which is on the potato chip packages, which I'm sure you all read religiously, and that's trying to remember what our recommended daily allowance. Okay, and that's the population number, but that's derived from a number called the EAR, an estimated average requirement, and that's some distribution in the population. If you're below the EAR, you're officially inadequate. But nobody seems to care, but it's not a pretty picture. So the American diet isn't a pretty picture. So vitamin, let's see, it's hard for me to.
You can walk over here, you got a lavalier.
Okay. So vitamin D, 70% of the population is below the EAR. You get vitamin D from sunshine. There's not too much in food. And you convert a cholesterol derivative to vitamin D. And then in the body, it's converted to a steroid hormone. It's just like estrogen in that how it works. It receptor recognizes it, and then it recognizes 12-base sequence in the DNA and either turns on genes or turns off genes. I'll talk about it a little later. But it's a really important hormone. And 70% of the population is low. Let's see, vitamin E, 60% is below the EAR. Magnesium, 45%. Now magnesium is in every DNA repair enzyme. When you damage your DNA, repair enzymes are cruising along the DNA looking for trouble and taking out the bad lesion and putting in the right base. But if you don't have magnesium, you're in trouble. And it's in 400 or so enzymes, and practically half the population is too low. Where do you get magnesium? Nature color-coded it for you. Magnesium isn't green, but it's in the center of the chlorophyll molecule. So anything green has magnesium in it. Eat a big plate of kale or spinach, you're getting your magnesium. You get a little bit from nuts, but so you don't eat those things and you don't get your magnesium. And then vitamin K, 35% of the population, calcium, 35%, vitamin A, 34%, vitamin C, 25%, omega-3s, 30% of the brain are these omega-3 fatty acids we get from fish. And you don't eat any fish, you can take linolenic acid, which is an omega-3 that's in some greens, in flaxseed and things that people don't normally eat, and convert it inefficiently to the omega-3s. But most of us are probably really low. They don't have an official number yet. And then zinc, I put as 8%. Zinc is in 2,000 different proteins. Need zinc. So there's zinc fingers that recognize pieces of DNA, and there zinc is in copper zinc SOD, which is an enzyme that destroys oxidants, and zinc is in P53, which is mutated in half of human tumors. It's very important for protecting you against cancer. And So if you just go between 10% of the population and 5%, you have a long list, which I haven't put in here. So the American diet isn't really up to snuff. Now, you don't give any one of these compounds to a rat, it'll die. That's the definition of a vitamin. But I'm trying to change the definition. I'll tell you later why. So what got me into nutrition is not that I just got tired of the field I was working in, but some scientific discovery sort of got me off into a new field. And I like to get into new fields, but nutrition's a very muddy field. It's a lot of, everybody told you, don't eat fat. And then everybody ate carbohydrate, and they found out the carbohydrate was worse. And then there were lots of advice that didn't turn out right. Now, they get some things right, of course, but I shouldn't knock nutrition too much, but it's a pretty muddy field. But I like to get into muddy fields because I come in from a different background, and sometimes I can make some contributions. Anyway, these are red blood cells, and they're the only cell in the body with no DNA in it. So every cell has a nucleus with DNA, because that's controlling synthesis of all the different enzymes. But red blood cells, the nucleus is extruded from the precursor cell, and you make a red blood cell with no DNA. Well, some scientist who was thinking out of the box once stained mouse red blood cells for DNA with a fluorescent reagent, and 1 in 2,000 mouse red blood cells lit up. It had a piece of DNA in there. What was that? And they tracked it down to when you extrude the nucleus, if there's a broken chromosome, it gets left behind. So it tells you there was a chromosome break. Now, the most dangerous part of radiation is when it breaks chromosomes. So this was a very nice, simple assay for measuring chromosome breaks. You irradiate the mice, you get more chromosome breaks. Well, anyway, a guy named Jim McGregor came to my lab on sabbatical, and he had just done an amazing experiment, which I'd like to show you. And he was using this test to see how does the cell repair these double-strand breaks, these chromosome breaks. And in the course of it, he had stumbled on something interesting, which I'll get back to in a minute. But in the meantime, he tried to get this test to work in humans, and it doesn't work at all. And the reason is that all your blood goes through the spleen, which is a little organ. Where's the spleen? Down here somewhere?
Opposite the liver.
Opposite the liver. Okay. I'm a Ph.D., not an M.D. So anyway, all the blood goes through the spleen, and the function of the spleen is to take out any blood cell that's a little stiff. It doesn't want to just clog up a capillary somewhere. Now, if you lose the spleen in an accident, you can live perfectly well. But when the spleen is working, you don't see this. And inbred mice that they were using for the mouse experiments don't have much splenic function, because in breeding them, they sort of lost that along the way. But they live pretty well. And people, so McGregor wanted to do this test in people, and it didn't work. So being very persistent, he went to Kaiser, which is our local health maintenance organization in California, and they have 6 million people in their database. So he said, do you have any people who lost a spleen in an accident? Oh yeah, we have hundreds of them. And so McGregor got permission to contact 20 of them, and he set up then he could see these micronuclei, these chromosome breaks in humans. And so he set up a baseline and found something very interesting. So it's a sort of complicated experiment, but I'll just walk through it to explain why what he did was so interesting. So this hatched area down here is the baseline. 19 people were down in this region without any interventions. But one guy was 20 times as high, and so McGregor thought that was interesting, and he followed this guy for a year, but he didn't know what was causing all this. But then in his mouse experiments, the RNA positive erythrocytes are also called reticulocytes, and those are the 1% of the red blood cells that have just been synthesized. They have a little RNA left in them, and you can pull them out with magnetic beads and look at them. So, and these are the regular red blood cells. So anyway, McGregor, in his mouse experiments, one day all his control mice were full of chromosome breaks. And he yelled at his technician, "Did you radiate them by mistake?" And she said, "Absolutely not." So he, instead of leaving it there, he tracked it down. And it turned out he called up the company that he tried lots of things, but when he called up the company that gave him the vitamin mix for the feeding the mice, they said, "Oh, Dr. McGregor, we're very sorry. We left folic acid out of the vitamin mix." Folic acid is a vitamin. Folia is the Latin word for leaf. And my mentor at Caltech, when I got my Ph.D. before all you guys were born, in 1950 to 1953. Anyway, before he came to Caltech, Herschel Mitchell had isolated folic acid from 4 tons of spinach. So foliage is the same root. And so they named the vitamin after the leaf. Okay, folic acid. So the only trouble is that they had oxidized the vitamin in the course of isolating it. It's very easily oxidized. And so from then on, they've been using this oxidized folic acid in vitamin pills, but the real folic acid in your body is tetrahydrofolate. It's a reduced form of folic acid, and it's one of the things I've been complaining about. Eventually, I think they'll go to a natural folic acid in the vitamin pills, but it's much more expensive. So anyway, that's a little side issue. But so then McGregor did a dose response on folic acid. The less folic acid, the more chromosome breaks. So it's just like getting irradiated. And so McGregor thought that was pretty interesting, and he went back to this human study, and he asked this guy about his diet. It turned out he didn't eat many fruits and vegetables, or salad or anything like that. And so McGregor gave him some folic acid, a folinic acid, which is a reduced form. And right away, as you can see, maybe, or you can't see, the reticulocytes, which are newly synthesized, came down in a few days, right down to baseline. It was a beautiful experiment. It established causality. Folic acid was what was doing this, and you give it, and it comes right down. It's one person. It's not epidemiology, but epidemiology is a very difficult science, and they get the wrong answer lots of the time because they don't understand mechanism. My epidemiologist joke is study shows Miami a weird place. Everybody's born Hispanic and dies Jewish. Anyway, so the red blood cells turn over slowly, and they came down, and then they ran out of the folic acid and both started going up again. Here's the retic. So McGregor said it's unethical to break this guy's chromosomes anymore. I mean, he's been doing it all his life, but anyway, so I better just put him on folic acid permanently, or get him to eat a better diet. So he went to the fellow and said, how about eating some greens? It'll stop all this chromosome breaking. The guy said, no, give me a pill. So he gave him folic acid, and he followed him for a year or something like that. And he stayed very low baseline, and then he moved out of town, and that's the end of the experiment. But he published this in 1988. And the great thing is he measured how much folate is in the serum and how much is in the red blood cells. And he could tell what level of folic acid broke chromosomes. So he looked up who in the population is at this level, and it was 10% of the US population. So people worry about this incredibly low level of radiation washing up on California after Fukushima. And of course, it was the tsunami that was killing everybody, because the tsunami is a 100-mile-per-hour wave that's 10 stories high or something, and it's filled with pieces of houses and wood and all sorts of things. That hits you, you're done. And so that's what killed everybody. But still, and that the radiation being diluted in the whole Pacific would really matter when it got to California seems crazy, but that's what people were worried about. Anyway, red blood cells have much higher levels of folate, as you can see or can't see, than the plasma. And everybody thinks red blood cells are only delivering oxygen to the tissue, but they're the FedEx trucks of the body. They're much higher in every goodie inside the red blood cell, and when it dumps the oxygen, it dumps all these other things. So, I don't know, that's an idea. I haven't proven it yet, but I think it's going to be true. But anyway. So, okay, anyway, so when I saw that half the poor were getting their chromosomes broken by not eating a good diet, I said, I'm getting into nutrition, because people were worried about parts per billion, pesticide, and other things that seemed all trivia to me. And smoking breaks your chromosomes, but you're getting a huge amount of material in you. Anyway, this is a slide from Fennick in Australia, who compared X-rays and folic acid deficiency in humans. And I had a French-Canadian postdoc, Chantal Courtemanche, who did the same experiment and published it about the same time, but I'll show you his experiment. Looks a little nicer on the graph, so I'll show you there his. So you don't eat your greens, it's like irradiating yourself. So don't worry about tiny, tiny levels of radiation, eat a good diet. Anyway, so I got very interested in micronutrients, and we helped McGregor work out the mechanism. When you get radiation, you get clusters of electrons. That's why radiation is bad for you, because it damages a base on each strand of the DNA. So occasionally you get this cluster of electrons, and this is an oxidized base, and the DNA, the two strands are wound around each other. But anyway, and then repair enzymes are all cruising along the DNA. If there's only one lesion, it clips it out and puts in the right base, and it fixes it. So 95% or more of the damage to your DNA gets repaired before it causes trouble. But if you have two nearby lesions, then they get repaired at about the same time, the chromosome falls apart, and that's the dangerous part. So anyway, we worked out a mechanism with McGregor. And so the next micronutrient I got involved with was a nutrition professor knocked on my door. I was in biochemistry. And he said, I'm an expert on iron. There are 2 billion women and children in the world who are getting too little iron, and it's wrecking their mitochondria. And you're an expert on the mitochondria, but what I want to tell you is the World Health Organization is going around giving iron to all these people, and iron's dirt cheap, but it's giving them too much iron. So too much iron is bad for you too. So why don't we get together and do an experiment on the whole range of iron from too little to too much and see what happens in rats. So we did that, and up is bad. And so we measured mitochondria pouring out oxidants, mitochondrial DNA getting oxidative damage, mitochondrial efficiency, all these things. Mitochondria are the little power plants in cells, and there are about 500 or so in every cell. And that generates the chemical energy, ATP, that powers your muscles and your brain and all your other biochemistry. So this red is the sum of all these things we measured. So this is 2 billion women and children, and this is a lot of old men who eat too much red meat. So, but this is the really important thing. This isn't nearly as much, and you should be in here, which gets to metals. metals look a lot like each other. Too much iron looks like zinc or copper. They have 2+ charges on each. And it's thought that too much iron interferes with copper metabolism or zinc metabolism, and that may be what's causing the trouble. Calcium and magnesium, one's above the other on the periodic table, and they look very similar. They both have 2+ charges. And if you take a calcium pill, but not a magnesium pill, you're flooding the body with calcium, and it can damage some magnesium enzymes that require magnesium, and vice versa. So metals you really should be careful about and don't overdo it. Mae West said too much of a good thing is wonderful, but I suspect she was thinking about sex, not micronutrients. So don't overdo it for minerals. So then the next micronutrient we looked at is David Killalay in my lab got interested in magnesium deficiency. So he took human cells in culture and made them a little magnesium deficient. And what he found is you got mitochondrial DNA cross-linking to mitochondrial proteins. So when you have radicals, when you get oxidation, you get radicals, and they can do things like cross-linking. So that, and he also showed that primary human cells go through a certain number of doublings and then they senesce, they get old. Well, if you make them magnesium deficient, they get old much sooner. Now, we don't know whether that's true for all micronutrients, but he just tried it on magnesium, and we published a paper. And they also cause telomeres to get shorter, and that's a measure of how many cell doublings and aging. Anyway. So then I started thinking about, why is nature doing this? Why is nature battering up our DNA? And I did a Google search. It was doing science before Google was a real drag, because you could spend the rest of your life in the library trying to find things. And it was just so much work and so much time. But Google, you just put something into Google, and out come all these scientific papers on the subject you put in. So it just makes science so much easier. Anyway, I put in the 30 vitamins and minerals, and then DNA damage and chromosome breaks and cancer, and asked what comes out. Well, there's a paper on calcium deficiency showing chromosome breaks by one guy, and then a paper showing cancer by somebody else. And then folate deficiency, several groups had shown chromosome breaks, and then somebody else, Willett, had shown cancer, and et cetera, et cetera. So there's a lot of stuff in the literature that when you made human cells or mice or people short of a vitamin, you started getting DNA damage. And that was my field at the time. I was very concerned on preventing cancer. So DNA damage leads to cancer down the road. And so I kept on mulling, why is nature doing this? And then one day I had an epiphany. I understand it. So that this has been happening through all of evolution. The soil doesn't have all the minerals in equal amounts. There are red soils with a lot of iron, and then some soils with no iron. Selenium comes in patches. So there are areas with too much selenium, and that'll poison you. And there are areas with too little selenium, and that'll poison you because you need selenium. So it's all so what I postulated is that every time you got a little low on one vitamin or mineral, what nature did is it did a strategic rationing. It puts it into those proteins that need that vitamin or mineral that are essential for survival and reproduction, and the proteins that are preventing long-term damage, some insidious damage like DNA damage or calcification of the arteries, those proteins got starved because it's down the road. So you're trading long-term health for short-term health. And it was perfectly plausible, and evolutionary biologists have used that idea for other things, but not for this. So anyway, I threw out that idea in a paper. And back 9 years ago. And then one day Joyce McCann, a senior scientist in my group, came into me and said, I'm a little skeptical of I call this system of rationing Triage, after the French battlefield word. And so Joyce said, I'm a little skeptical of your Triage idea. I think there's a better way to tackle it. I said, Joyce, what do you want to do? Go to it. And so what she did is she looked over the vitamins and minerals, and she go over here this time, and picked ones that were well studied but not too complicated. Magnesium is in 400 proteins, zinc is in 2,000 proteins. You'd spend your rest of the life in the library. So she picked vitamin K, which is in 16 proteins, and then she picked selenium, which is in about 25 proteins. So they were complicated enough, and there was a big literature on them, but nobody had ever looked at the literature from this point of view. Is there a rationing when you get low? And she wrote 2 beautiful reviews, and both of them fit that idea. It's built into cells. So I'm convinced it's a general thing, but people should show it. So I'll just go over a little bit about vitamin K. Vitamin K is a vitamin that was discovered by a Dane named Dam and got a Nobel Prize for it, that there's a compound in plants that is necessary for blood clotting. So you have to eat that chemical, otherwise your blood won't clot. And what it is a compound called phylloquinone. It's used in photosynthesis in plants, and what animals have hijacked it for is to be a cofactor for an enzyme that makes a protein bind calcium. So an amino acid called glutamic has an acid group on it, and with this enzyme and a cofactor, you can put a second acid group, so it has 2 acid groups, and it can bind the 2 positive groups on a calcium. So all these 16 proteins are calcium binding proteins, and the blood clotting net is calcium protein. And those are the essential ones, because you cut yourself pretty often, once every couple of weeks or once a month, or shaving or whatever. And if you didn't have blood clotting, you'd bleed to death. So what Joyce McCann did is she looked at the mouse knockouts. Kopecky got a Nobel Prize for figuring out how to knock out any gene in a mouse. And that's been a very valuable tool, because you can knock out a gene and ask what happens. Well, you knock out any of these blood clotting genes, they're embryonic lethals. They never get born. And because you're making tens of thousands of kilometers of capillaries in a mouse or in a human, I don't know the exact number, but it's something like that. And if there was any little imperfection in making all this, the animal would bleed to death, and you never get a live animal out of it. So she said those are essential. It's a somewhat crude measure, but those are clearly essential for survival. And people cut themselves pretty often. And the others are the ones we're really interested in. Those are the proteins that you disable to help these proteins. And so osteocalcin is in the bone, helps boost calcium around the bone, helps make strong bone. Gas6 protein has to do with diabetes, and I won't talk about that. Matrix Glau protein is in the blood and prevents calcification of the arteries. And TGF-beta inducible protein, they knew about this protein, but they only found out that it had this funny amino acid Gla in it, which is an abnormal amino acid, so they knew it was vitamin K dependent. And those animals are born, they look okay, and then they all drop dead of cancer. And they tracked it down to its interacting with the microtubules, which separate the chromosomes at mitosis when the cell divides. And if you don't have the protein, you lose a chromosome here, or you lose a chromosome there, and that leads to cancer. So you don't get an accurate mitosis. So all this was, and periostin is, again, something to do with diabetes. So all these things have medical interest because, so what Joyce did is she looked at the whole range of things. So here, osteocalcin, its function is to make strong bones, and you look at the mouse knockout phenotype. The animals are born, they look okay, but you stress them and the bones break. And in humans, you look at the human gene that you knock down in a mouse, and you have bone mineral density is bad. And then you look at 30 million people take warfarin or Coumadin. Anybody here take warfarin or Coumadin? Anyway, it's a very common drug. And they give it to you when you have too many thromboses, and that can kill you. And this interferes with vitamin K and prevents this blood clotting. But what they're doing on the side is disabling all these enzymes that are the long-term enzymes. And so bone health is shown is a problem in people taking these drugs. And then vitamin K deficiency, it's bone health. And matrix cloud protein, the animals live, they're born, they live okay, and then they drop dead of heart disease, of calcification of the arteries. So what this protein does is prevent calcification of the arteries. And one theory about this is calcium phosphate crystallizes very easily and makes crystals. It's very insoluble. And they think that, and there's a lot of calcium in the body and phosphate in the body. So if it comes out in the arteries, then it's the center of an atherosclerotic plaque. And so this protein prevents that. So all these kinds of proteins that are keeping you with a long lifespan, it's all insidious damage that builds up, get trashed. And again, it fits everything. So the people who get anticoagulant therapy have a much higher rate of dying of calcification of the arteries, and vitamin K deficiency gives you. And this protein turns out to be turned on by vitamin D on top of all this. So there's another input there, but I won't go into that. So anyway, we got all excited by this and decided really we should improve micronutrient status for people. Micronutrients are dirt cheap. You can get most of them in a multivitamin pill, but the people who take multivitamin pills are the people eating good diets, and the people who don't take them are the poor eating terrible diets. So and then Joyce did another review on selenium, and again, it's mortality and cancer and DNA damage and heart disease and immune function and muscle and cognitive function, all these long-term things in the ones that are being robbed of or not given the selenium because it's going into the proteins that are essential for survival. So it makes sense, and these are two good examples. It takes about 9 months of just using Google and going to the library and reading 500 papers to do one of these reviews, and she did a beautiful job. And so how does the system ration? Well, in the case of vitamin K, it's a little complicated. The vitamin K is converted to something called menaquinone. First of all, when you eat plants, the phylloquinone goes right to the liver, and you do only the coagulation proteins in the liver. But these other proteins are done in the other tissues. So how do you do those? Well, when you send the vitamin K to the liver, after it's done its job, it gets converted to something called menaquinone, which then goes into the plasma and goes to all the tissues equally. So then you can do all these other proteins. So it's a way of doing it. You could do it by binding constants and other ways, but that's how vitamin K does this rationing. And then selenium is the other one, uses a completely different system. So selenium transfer RNA puts the amino acid into protein, and in the case of selenium, a selenocysteine is done in only these few proteins. You put a selenium in instead of, selenium is very much like sulfur, except it's a stronger antioxidant, and so it's used for particular purposes. And it's a modified base in the transfer RNA that puts this in that gets damaged. I won't go into all the chemistry, but it's, both systems have this rationing. So what is the advantage of this kind of analysis? Well, one, it gives a mechanism how moderate vitamin mineral deficiency increase risk of a disease of aging, all this insidious damage that's helping you have a long life, and suggests a prevention strategy. And two, what we found is about half the proteins are essential proteins, and half the proteins we analyze are these longevity proteins. So I coined the word longevity proteins because they're involved in giving you a long life. So should a vitamin just be something that when you don't give it to a rat, the rat dies? Or should it be something that gives you a long, healthy lifespan? I think most people would pick the latter. So I just want to redefine vitamins, and that suggests that they're going to be new longevity vitamins and minerals. And in fact, I'm writing a review about that now, and I'll give you one example. But it also says that what we should be doing is measuring one of the proteins that is vulnerable, that goes first. And I have an entrepreneur friend, scientist entrepreneur, who can put a finger in and take a finger prick of blood and measure 1,500 proteins in a finger prick of blood. Analytical methods are getting terrific now. And I have another entrepreneur scientist friend who put a machine in every hospital in China that'll measure a couple of dozen proteins of medical importance. So these things are coming fast. The Chinese subsidized him, and he didn't want to fight FDA for 10 years before he gets something in. So he just did it in China. And so now that it's successful in China, he'll try and sneak it into the US. But FDA wants control. They don't want you determining your own health and cutting out the doctor. So it's, anyway, that's my somewhat cynical view, which you don't have to believe. Steve would probably think differently. Anyway, I'd like to say a few things about obesity. The conventional wisdom on obesity is it's too many calories in and too little exercise out. But I've been thinking differently for a while, because when you analyze the diet of the obese, and there are 4 or 5 papers doing that, they're eating the worst diet in the country, if you have to find worse, as ratio of calories to vitamins and minerals. They're eating white bread and white rice and white potatoes and things that don't have any vitamins and minerals in them. And so the obese are eating this god-awful diet, and that makes them get fat. Not every fat person is that way, but that's what the numbers say. So this is calories in, and this is exercise out. So now exercise adds a couple of years to your life, and it's a good thing. My wife is Italian, so she feeds me a wonderful Mediterranean diet, but she kept on nagging me, I'm spending too much time in the lab, and I'm not getting enough exercise. So one day I said, when I feel like exercise, I run my experiments, I skip controls, and I jump to conclusions. So I liked that joke so much, I told it 50 times, and my wife said, I've heard enough of that damn joke. I'm getting you a personal trainer. So now I go twice a week and row and lift weights, and I'm sure it's good for me, but it does take time. Yeah. Anyway, so maybe this is the average diet of 20 to 30-year-olds. These are the main energy sources. Sugary soft drinks, number 1. At least there are a few veggies on here. Potato chips and popcorn. It's not so it's not a very healthy diet. And the obese are even worse. So I got together with Mark Shiganaga, who's a scientist in our group, who's the expert on gut. And we said, hey, we have to get, he's very worried about fiber, because fiber is very important for gut health. And vitamins and minerals, I argued, that are important for your longevity. So we said, how do we get these into the poor? And so obesity is really a disaster in terms of health. Every disease of aging that's been looked at is accelerated in the obese. Cancer and heart disease and cognitive decline and diabetes, you name it, if it's been looked at, it's much higher in the obese. And I think in part that's due to the fact that they're eating a bad diet, not just eating too many calories. And maybe when you don't get your fiber and don't get your vitamins and minerals, it makes you hungry. There's a little evidence for that for calcium, but we don't really know whether it's true or not. But anyway, so Mark and I decided to develop something. And pills are out because the poor aren't known to be reliable on taking pills. And you couldn't get fiber into pills, and there'd be too many pills they'd have to keep track of. So the local USDA has the job of taking California fruit that's not being used and figure out new ways of using it so it adds to the California economy, and that's what they do. So if a fruit gets blemished, they can cut that off, but what do you do with the rest of it? You can't sell it. And so they made a fruit bar as a snack of all this extra California fruit that's dirt cheap. And it worked, and they got a company to sell it. So we heard about it and went over to the USDA lab, which is very near Berkeley, and knocked on the door and said, hey, could you add some vitamins and fiber to this bar? And they said, sure. And we worked together, and then we added a few other ingredients, and we tried to put all the ingredients that are in a Mediterranean diet that the poor weren't eating, the obese weren't eating. And it took us 10 years and 18 human clinical trials to do this, but we finally got there. I did it all on a shoestring, actually. So.
Shoestrings taste good?
Yeah. You know what one shoelace said to the other? One said, you want to go for a walk? And the other one said, why not? I learned that in grade school. Okay, so this is our first paper on this. We call it the Corey bar, because I work at Children's Hospital of Oakland Research Institute. And I used to be a professor at Berkeley, but I retired from there. And I'm 86 and still not retiring, so I told a colleague I'm doing the best work of my career, and he looked at me and said, Bruce, you've been telling me that for 30 years. So, one more joke. I used to work on mitochondrial decay and aging. Mitochondria are the power plants in cells. And when I turned 86, I decided my mitochondria are decaying ever more rapidly. But I finally figured out what they're decaying into: hypochondria. So these are 25 people in our clinical trial, and we're measuring HDL. HDL is the good cholesterol. It's actually, cholesterol isn't involved at all, but it has to do with size of these lipoprotein particles, but that's new development. So you shouldn't worry about cholesterol. People told you not to eat eggs. Steve Zisel here has been saying eggs are really good for you, and et cetera, et cetera. But anyway, so at the bottom of each arrow is where we started before we gave them the bar. We said, don't change your diet, just eat what you're eating and take 2 bars on top. And these are lean people around Corey, mostly technicians or scientists who think they're eating pretty good diets, but we could raise practically everybody's HDL in 2 weeks.
Wow. And that's good.
That's good.
That's good.
So we said, wow.
Does it affect
I'll get to that. Yes. But, and then we had blood samples from all these trials, and Joyce McCann, who did those reviews, is terrifically organized, and I'm terrifically disorganized. So I said, why don't you run this project? So she became project leader on the Corey Bar and really did a wonderful job on it. So we just published this paper, just came out, published on the web, a multi-component, that's the second paper, a multi-component nutrient bar promotes weight loss and improves dyslipidemia, which is HDL and LDL, and insulin resistance in the overweight obese. Chronic inflammation blunts these improvements. And I'd just like to show you. So the bar does wonderful things to people. And we told them, don't change your diet. So or at least we didn't give them any instructions on changing their diet. And people have been trying to get the diet of the poor changed for 20 years, and they've not been very successful. There have been many studies that really were very disappointing. So this is one of the first studies we did. Here's in the overweight obese. One, we had to do 2 months because the HDL going up in 2 weeks in lean people doesn't work in obese people. Everything takes longer. The metabolism's all fouled up. So, but we got HDL going up, but triglycerides went down, and blood pressure went down, and
Heart rate.
what was that?
Heart
rate went down, and then glucose metabolism, insulin resistance went down, which is a big thing, and then my weak eyes at my age.
That's insulin.
Insulin went down, glucose went down, inflammation went down, and though it wasn't statistically significant, there's a lot of different range of inflammation, so there's so much variability, it wasn't statistically significant. And then on weight, there's a hint something's happening, and waist circumference looked like something was happening, but not much. But then all the new knowledge in heart disease isn't interested in cholesterol, it's particle size. And Ron Krauss, who's a clinician heart guy in the lab next to mine, is the world's expert on lipoprotein. So I knocked on our door and said, why don't you, we have all the blood, why don't you measure the particle size? And he did, and it's beautiful. It's just like statins, only cheaper. So this is looking now at the whole population of HDL, and then these are the higher risk particles, they're all going down, and these are the lower risk particles. But a lot of things, some of them weren't statistically significant. But then we did another experiment. If you look at CRP, which is a measure of inflammation, you can see that there's a tremendous variability. So what we did is we divided the population into those less inflamed and more inflamed. Now this is a smaller number, so you'd think it'd be less significant, but it was much more significant if they're not inflamed. So this is the lower inflammation, and as you can see, very good statistical numbers, and weight loss became really appreciable, and waist circumference. So you can get the overweight to lose weight, but they have to be not inflamed. So inflammation blunts all this. So, and this is the inflammation. So I know you guys can see it, but anyway, all these good things were statistics. So we're really improving the metabolism. And when we brought it to Ron Krauss, he said, wow, better than statins. Now, I don't know whether he wants to be quoted on that. But anyway, and statins are a $35 billion industry, and this is food. So I think the future of prevention is nutrition. That's what we're not eating well. And I think it's going to help in obesity, it's going to help in cancer and all these diseases. But we have to do the work. And Anyway, so, and I'll finish off by just showing you, I got a new postdoc in the lab, Rhonda, about 2 years ago, and she's a tsunami coming through the lab, full of energy. She has a blog with 20,000 followers or so called foundmyfitness.com, and she does a video on nutrition every couple of weeks. And plus she's been doing experiments in the lab, or has a group doing experiments, and did 2 beautiful theoretical papers that I think are very important. So I had known that autism is related to latitude. In northern latitudes, they have more autistic kids. And people said, that's probably vitamin D. Because vitamin D is ultraviolet B light from the sun, and the more north you go, the less sunshine on your skin. And it's also related to skin color. But I read somewhere that tryptophan, the amino acid, was related to autism. Some people published a paper. And tryptophan goes to serotonin, which is the main social hormone. So autistic kids don't look you in the face because there's so much information, and serotonin controls sensory gating. So you just don't have enough bandwidth to get all that information, so autistic kids don't look at you. Anyway, I asked Rhonda, can she figure all this out? Is there a vitamin D connection? And vitamin D goes to a steroid hormone, and I'll just like to say a few things. So vitamin D hormone is from ultraviolet B light. Those are the burning rays of the sun, but they convert a cholesterol derivative to a precursor of vitamin D. And then the body converts the vitamin D to a hormone just like estrogen. And that hormone binds to a receptor. And so the ultraviolet B light converts 7-dehydrocholesterol to vitamin D3, which then in the liver gets converted to 25-hydroxy vitamin D3. That's what they measure to see your level. And then the kidney converts that to this hormone. Now that hormone controls 1,000 genes. It has to be important. You can read that sequence of DNA bases in the hormone that the vitamin D binds to receptor and binds 12 bases. And you can just look through the genome and see what genes are controlled. Well, it's over 1,000 genes. And tryptophan, which is one of the essential amino acids, that gene that puts a hydroxyl group on tryptophan hydroxylase, is controlled by vitamin D. And that's the precursor to serotonin, which is this important social hormone. So what Rhonda found is that there are 2 genes making serotonin, one in the brain and one in the gut. Now the one in the gut is a more powerful enzyme, and this is 6 bases, 3-base spacer, and it's 6 bases. And both these sequences were a vitamin D sequence. That's all in the literature. So she looked and found this in the front of each of these genes. But the sequences were a little different, and instead of stopping there, she looked in the literature, and someone on a different enzyme has shown that some of the sequences turn off the gene and some turn on the gene. It depends on the sequence. And so this, if there's a T at this position, it means it turns off the gene. So the vitamin D hormone is activating the gene in the brain, but turning off the one in the gut. And if you get too much in the gut, it's bad for your gut. So it was more complicated, but it explained all sorts of things about vitamin D that nobody had realized before. So this shows how vitamin D hormone works. The vitamin D receptor, and vitamin D binds it and then finds this, takes a partner and then goes to the DNA and binds it there and stops that gene from being translated. So and that means, or turns on the gene, lets it be translated. This sequence turns it on, and then and this sequence turns it off. This is all theoretical. It was all lying on the ground in Google. It's just a matter of asking the right questions. And so anyway, she published this paper, and I think it's a landmark paper. It didn't get much press release because it was a review, and they don't normally cover reviews, but it was all theoretical. So I was very proud of what she did. And then it explains all these characteristics that people associate with autism but didn't understand why. So, one, people had found low levels of serotonin in the brain and high levels in blood cells. And they didn't understand why, but this explains why you're turning off the ones outside the brain. And serotonin doesn't get through the blood-brain barrier. Tryptophan will, but not serotonin. And psychologists have done these scary experiments. What they do is there's a trick for lowering serotonin in ordinary people's brain. You can give a batch of branched-chain amino acids which interfere with tryptophan, the precursor of serotonin, and you can lower serotonin by 80%. And you do it to ordinary people, they become aggressive and hate everybody, plus they get completely impulsive. They lose all the ability to do way alternatives, and they just get very impulsive. And these kinds of behaviors are known in autistic kids. There's a huge spectrum, so it's not everybody. So, and then boys get it much more than girls, and it turned out estrogen had been shown to turn on the one in the brain. So estrogen sequence is somewhat similar to the vitamin D sequence, and this particular gene can turn on by either. So if you have estrogen, you don't need the vitamin D, but if you don't have estrogen, you do. And so that explains why boys get it at 5 times the rate of girls. And then association with vitamin D. Okay, I won't go into these last two. But anyway, it explained a lot of things about autism that weren't understood. So we thought, the theory must be right, and everything fit the biochemistry. And since we've published this, several papers have come out or been submitted that refer to our paper and confirm the biochemistry and confirm other things about it. Now, there haven't been a lot of clinical trials on vitamin D and on omega-3 fatty acids that are in the brain are also important. So, but everything so far looks like it's really going to be right. And it opens up prevention. Because if you have genetic defect, it's hard to intervene without knowing how it works. But the biochemistry tells you what the players are, and you can try them. Now, if the mother is low in serotonin, you may not be able to reverse all the changes, but there's small studies showing that maybe you can, and it's worth trying to prevent it and maybe to modify it. Now, so this is, these are all the things that serotonin controls. It controls social behavior, impulse control, sensory gating, decision-making, emotion, aggression, anxiety, all these things. There's a whole literature on serotonin, and now we know that vitamin D controls it, and that omega-3 fatty acids are important for its function. So, now Rhonda's second paper just came out, and Rhonda Percivalli-Patrick. And so vitamin D and the omega-3 fatty acids controls serotonin synthesis and action, relevance for ADHD, bipolar, schizophrenia, and impulsive behavior. And this, again, looks at the literature. And, okay, so I'm getting right to the end. And there are all sorts, I won't go into skin color because it's a very touchy subject, except to say that if you have a dark skin, in the tropics, it protects you against burning by the sun. And if you put an Irishman in Australia, they're in trouble. And the solution is a hat and sunscreen. Now, if you put an African American in Chicago or an Indian who has a dark skin, they're in trouble. And the solution is a pill that costs a few pennies of vitamin D. So genetics isn't destiny. Genetics controls your skin color, and you can see why the tropics, you need a brown skin so you don't get burnt to a crisp and eaten by a lion and taken out of evolution. But one kind of understands it all, and you can do something about it if you understand it. So I'm very optimistic that all these micronutrients are involved with so many pathways. Practically every pathway has a micronutrient in it, and you're disabling pathways right and left if you don't eat a good diet. Now, some of it may be reversible. If you then start eating a good diet, you may reverse the change. But in DNA damage, you don't reverse it. That's irreversible. So if you have DNA damage and it gets you into a mutation, you have it. There's no way of repairing it. Anyway, I just like to, this is Rhonda's slide showing that to release the serotonin from neurons, EPA, which is one of the two omega-3 fatty acids in the brain, is necessary to antagonize the E2 prostaglandins, which inhibit this release, and EPA reverses that so you can release it. So it's important to get these fish compounds in your brain. And then membrane fluidity is controlled by DHA, another one of these marine fatty acids, and that's important for action of both dopamine receptors and serotonin receptors. So the three social hormones in people are serotonin, oxytocin. Oxytocin's a peptide hormone that bonds the mother to a baby. When she has a baby, it goes shooting up in women and bonds to the baby like glue. That's its purpose. And women have higher levels of serotonin than men. They have card games that the economists have that can measure your trust level. Women are more trusting than men. And but vitamin D is controlling that oxytocin. And they've done you can't get it in through the blood-brain barrier, but if you spray it through the nose, it's sort of a back door to the brain. So you can spray serotonin into the nose of men, and they become more trusting in these games, more like women. So maybe we should have bombers spray oxytocin in the air over the Middle East or something. Okay, that's all. These are the people in the group. This is Rhonda, who I think walks on water, but she was a surfer, and she put herself through college by teaching surfing. Anyway, and there's Joyce McCann, who ran this Koribar project, and there's Jung Soo, a Korean background who's brought up in Thailand. He's our metabolomics guy, and he's added to everything. Here's Mark Shiganaga, my Buddha. He looks a little like Buddha. And these are the two physicians that were involved. Michelle Mita Snyder is now co-director of the obesity section. She's an MD pediatric cardiologist, and she moved to Washington. Her husband's a physician too and got a job there. So she's the co-director of the obesity center at Children's National Hospital in Washington, D.C. And Ash Lau's a pediatric hematologist, and he's running our clinical trials now. Thank you.